Hi, my name is Wes Thompson, and I'm going to give a lecture on the genetic component of the ABCD study. So let me share my slides. Let's see, full screen. Share my slides. Okay, so again, I'm going to talk about the ABCD genetic data. I'm Wes Thompson. This is actually a lecture created by Chun Fan, who's the chair of the genetics work group in ABCD, and he's also my co-director at the Population Neuroscience and Genetics Center at the Laureate Institute for Brain Research, where we both reside. Um, Okay, so first, an outline of the talk. So I'm going to talk about the types of genetic data available in ABCD. So the probably the primary one of interest here is the single nucleotide polymorphisms and the genotype data. I'm going to talk about the genotyped versus imputed SNPs, and then talk a little bit beyond SNPs, including the twin component of the data, which is another way to assess genetic information, at least indirectly. I'm going to talk about special considerations for the genetic analysis of ABCD, including its diverse background population, and also enrichment for relatedness. So there's lots of siblings. There's monozygotic and dizygotic twins, and so forth, and that causes issues with doing genetic analyses. And then I'll give examples of using genetics in ABCD, including for genome-wide associations using the SNP data, and also, as well as polygenic risk scores. So first of all, the ABCD genetic data, the primary data of interest here are single nucleotide polymorphisms, or SNPs. These are genotyped through the Thermo Fisher smoke screen arrays. There's data on 11,668 participants. The genotyping array genotyped 516,382 SNPs. And there's many different things you can derive from these SNPs, some of which I'll talk about in later slides. So first of all, so what is a SNP, or what is a single nucleotide polymorphism? There are locations in the genome that have significant amounts of variation in the population. So for each location, you can have a GC or a TA. G is always paired with, these are the DNA sequence base pairs. So G is always paired with C, and T is always paired with A. And then you also, and so for a given location. So for example, here's person one, person two. They have the same base pairs up to where the one on top has a C, and the person at the bottom has a T. And so this is a place where there's some variation in the base pairs. And so this is, if this happens with enough frequency in the population, this is called a SNP. And so this is essentially the unit of analysis for the SNP data, is looking at like who has variance at specific locations. Um, and remember, you inherit two pairs, a pair of chromosomes, one from your father, one from your mother. And so often what happens is then you count the number of genetic variants at a given locus. So if this person had, say, person two had a T inherited from their father, but a C inherited from their mother, then they would have a count of one. If that person had two T's, they would have a count of two. And if this person had both C's in that location, they would have a count of zero. So with respect to the reference allele, which is C, this person would have zero, one, or two. And so the SNP data consists of a locus in the genome, a reference allele, and then the number of counts of the variants from that left reference allele, and that could be zero, one, or two. And so here you can see in the bottom right, you can see kind of a schematic of how this might work over many people. And so there's so the if from left to right, there's one, two, three, four, five people with a variant at the first location. There's one person with a variant in the second location, one person with a variant in the third location, so on and so forth. So this is essentially the units of analysis. you have over 500,000 genotyped locations in the genome with this smokescreen array. And at each location, everybody's given a count of 0, 1, or 2 with respect to how many different how many variants they have at that location. So, and then the very simple thing to do then is to associate that count with a phenotype of interest, a behavioral phenotype or some physical phenotype or something. So, and then you do 500,000 separate analyses, and that's the basis of a GWAS, of a genome-wide association study. So there's many more than 500,000 SNPs in the actual population. There's millions and millions of them. So it seems like by genotyping 500,000, we're ignoring a lot of information. So because the genotyping array is sparse. But the interesting thing is that there's a very strong correlation structure in human genomes due to the fact that the genetics from the meiosis that they're inherited in blocks, and so that creates a kind of a block correlation structure in the population. And so it turns out that you can actually impute the a lot of the SNPs that you don't assess. So you can actually using a reference panel. In this case, the next genetics work group in ABCD used the top med reference panel, which has some population diversity built into it, which makes it more useful for imputing data for in a diverse sample like ABCD. But so using that imputation procedure, you can basically bootstrap from 500,000 SNPs up to millions of SNPs, and it's roughly 10 million SNPs that have been imputed in the ABCD sample that's been released. So in fact, you can have like 10 million locations in the genome that you have pretty good information about what the allele count is for the variant, and you can do 10 million associations in a GWAS, and which is a lot, and that's kind of but that's much more powerful than just relying on the 500,000. So beyond SNPs, there's a lot of other types of genetic information you can get from genotyping arrays beyond SNPs. So there's copy number variation. That's where you have parts of the gene in the genome where you have like either additional genetic material or deletions. So you can have like deletions or duplications at certain areas, like 22q11, and those can have impacts on phenotypes. You can have whole genome sequencing. That would be essentially, instead of imputing 10 million SNPs, you could actually like measure the variation across the entire genome, which is several billion base pairs. You can create genetic instruments like polygenic scores, which I'll talk about. The copy number variation, the CNVs have not been released. The whole genome sequencing, a good chunk of this ABCD sample has been whole genome sequenced, but those data are not yet released. They will be in the upcoming data release. And then polygenic risk scores, those polygenic scores you can already compute, and several papers have been published using those. I'll talk more about that in a later slide. So for this talk, we're focusing on what we can get from SNPs, as like I said, this is the main data output currently available in ABCD. So genotyped and imputed SNPs, which I just talked about. I'm going to talk about SNPs, the derived genetic ancestry from SNPs, so genetic principal components, which is an important concept to understand. I'm going to talk about how SNPs have been used to derive genetic relatedness, because there's siblings and half siblings and cousins and stuff like that in ABCD. And then I'll talk about how to derive genetics risk propensities or polygenic scores, PGSs. So first of all, diverse population. The population, the sample of ABCD reflects the population diversity of the country. Not perfectly, but to some degree, like it's diverse in the way that the United States is diverse. And so here, you can see on the bottom, you can see essentially a plot of the proportion of genetic ancestry derived from different continental ancestral groups. So you have the African, East Asian, European, and Indigenous American, and you can see that there's there's a good chunk on top. You can see a good chunk of people who have mostly European or solely European ancestry, but there's also a good chunk of people who have admixture, so African, East Asian, and Indigenous and American. And so this is an important thing to consider when you're doing analyses because genetic ancestry can create confounds. So that because people of different genetic ancestries also in the United States are sorted into different environments. And so if you don't account for that, it'll look like the entire genome is related to certain phenotypes, when in fact that's a spurious relationship. Okay, I'll talk more about that in a minute. So the idea here is that this is called population stratification. So if you have people of different genetic ancestries assorted into different environments, so the population structure, for example, and on the left panel, can be related, it can be correlated with imaging features and also behavioral outcomes. And so this can create spurious relationships between imaging features and socio-behavioral outcomes. If you control for the genetic PCs, the principal components of genetic ancestry, which is the right panel, then that basically controls it gets rid of this spurious correlation between imaging features and socio-behavioral outcomes. So essentially, population structure is a confounding factor in analyses, even when genetics is not the primary like subject of interest. So genetics can be useful even in situations where genetics is not the primary unit of interest. So here's kind of a simple demonstration of the concept of population stratification. So there's something called like the informally called the chopsticks effect. So if you did a GWAS of using chopsticks, which is silly, it's a ridiculous thing to do. But if you did that in a place like say San Francisco, you would have very strong genetic correlations between genetics and using chopsticks. And the reason is quite simple, is that people of East Asian ancestry tend to use chopsticks, and people of European ancestry tend to not use chopsticks, though that's changed over the years a bit. But you'd still find associations across the genome where the allele frequency has drifted across populations from genetic ancestry. And that would create a spurious relationship between genetics and using chopsticks, it's completely obvious that genetics has absolutely nothing to do with using chopsticks whatsoever. So if you don't control for genetic ancestry in this case, then the entire genome would look like it was related to a behavioral trait. So it's important to consider this genetic ancestry in ABCD whenever you're doing genetic analyses. And it can be important, like I said before, it can be important even in analyses where genetics is not the primary concept of interest. So, and just to point out like things that might be obvious, but just to be clear about this, race, ethnicity is a social construct. Genetic ancestry is a genetic trace of the population lineage. They're correlated with each other, but they're not at all identical to each other. And so it's easy to conflate race, ethnicity with genetic ancestry, but they are not the same thing. So just to be clear that when I'm talking about controlling for genetic ancestry, I'm not talking about controlling for race or ethnicity. I'm talking about controlling for allele frequency differences of people who have ancestries that originated in different parts of the planet. Another aspect of genetic analyses in ABCD is enriched familial relatedness. So there's a number of there was intentionally an enrichment for twins, and so there's 422 monozygotic twin pairs. There's 659 dizygotic twin pairs, and there's 845 other sibling pairs. And so there's a significant chunk of the ABCD sample that has is genetically related to each other. And you can assess that via their genetics. You can assess how related they are to each other via the genetics. So if they have a if they're essentially 100% related to each other, so they're on the right end of this plot here, they would be monozygotic twins. They should like monozygotic twins should essentially have all their whole genome related to exactly the same, but like because of like genotyping variation, like it might not be exactly 100%. But anybody on this right like bump here is going to be a monozygotic twin. In the middle here, where it's between 0.4 to 0.6, that's kind of the range at which you would see full siblings. So that would be the dizygotic twin pairs plus the other sibling pairs, and then the rest of it, could be like maybe there's like cousins, first cousins, second cousins, and people who are completely unrelated to each other. So that's something to consider when you're doing analyses in ABCD, because people who are more genetically related to each other tend to be more similar to each other in other ways. And that's something that you have to count for either as a nuisance characteristic of your analysis by including random effects of family or something like that, or something that you can leverage, for example, in twin analyses for estimating heritabilities. So, relatedness, like I was saying, relatedness can bias your results. So, twin siblings share environmental factors. They also share, of course, genetic factors. Monozygotic twins share 100% of their genes, and dizygotic twin siblings share, on average, 50% of their genes. And if they live in the same family household, they also share a significant chunk of their, they have a significant common shared environment. And so those things make them more similar to each other in with respect to physical and behavioral characteristics than people who are not related or don't live in the same family or household. And so those are things you need to be careful about when you do analyses. Like, for example, doing mixed effects models with a random effect of family would be one way to try to at least partially control for this type of relatedness. Um, you can also going back to how to use the genetics data for scientific investigations. So here's something from a paper from Rob Lofman in Neuroimage in 2022. You can do essentially GWAS, or genome-wide association studies of different characteristics. Like, for example, here is one looking at the brain morphometry. So you can essentially do like associations for across the brain for with the SNPs, and try to figure out what are the genetic correlates of brain phenotypes. The ABCD, despite the fact that it's a large sample for a neuroimaging study, is actually very small for a primary GWAS. And so those can be several hundred thousand or even millions of people sometimes. And so because the GWAS typically takes a large sample to be well powered. So for a lot of phenotypes, I think ABCD will be underpowered. But not, I mean, but you can still, like, there's still been, significant results found using GWAS, like using significant, like the 5 times 10 to the negative 8 threshold, which is the usual threshold for significance for SNPs. But in general, it's not going to be like very well powered for primary GWAS. It could also be used for replication studies. So if you had a very well powered GWAS and then you wanted to replicate, you could use the ABCD data to do that. And then you can also, like, a very common use for the genetics data is to create polyvertex scores, which I'll talk about in a minute, how to create those. Poly polyver so polyvert You can use them to create polygenic risk scores, but you can also use this to create polyvertex scores, which are aggregating effects across the cortex using weighting across the vertices. And that's another way to increase power. If you're interested in that, take a look at this Lofman paper for more details. So polygenic scores, getting back to that. So the idea here is that for complex traits, like most behaviors, like neurocognition or mental health traits, tend to have effects that are widely distributed across the genome. So any individual genetic locus, any individual SNP will have a very tiny effect. But if you add them up across the entire genome, you might have a more substantial genetic effect. And so the idea is what you do is you train or you get summary scores from a GWAS that has been trained on a very large sample, so it's well powered to get individual weights from beta weights across the genome. So beta J is just the weight of an association analysis of SNP J with a given trait. Let's say like height, just to be simple. And so you can have millions of these beta J's. And they've been trained on a very large sample. And what you do is you take those summary statistics and then you apply them to the genetic data in ABCD. So XJ would be the 0, 1, or 2 count for a SNP for a participant in ABCD. and then you weight that by the relevant weight beta J from the training sample, from the summary statistics, the training sample, and then you just sum that across the genome. And so your predicted phenotype, like here, your predicted height based on genotype for a person in ABCD would be this weighted sum of their genetic variants, where the weights are the betas from a well-powered GWAS, external GWAS, and that creates one genetic prediction per person per trait, and that tends to be much better powered than trying to do like GWAS on individual SNPs in ABCD. So, and then you can use those polygenic scores for prediction analyses. Like you could very simply in height, you could use the genetic prediction of height to predict that person's actual height. That's not a very interesting analysis, but that's something you can do with it. More interesting might be like if there was a GWAS of, say, a personality trait, and you use that to predict mental health outcomes in ABCD. So here's an example of a polygenic score also by Rob Lofnan. And so here he had done polygenic scores. If you look across the bottom for different mental health traits like ADHD, autism, bipolar disorder, depression, so forth. And then he used that to associate, he used those polygenic scores to associate with different phenotypic traits, behavioral traits that have been measured in ABCD, like the CBCL, KSADS, and the UPS and the NIH toolbox. And you can see based on this heat plots here that there's a lot of different significant associations. So, for example, like a polygenic score, if you look on the very left, on the very bottom, if you look at the polygenic score for ADHD, that was very highly positive, significantly associated with the CBCL total problems as assessed by the caregiver. So you can use these genetic instruments, these polygenic scores, as means to investigate the genetics of traits in ABCD. And like I said, that's much better powered than just trying to do a GWAS of that trait using just the data in ABCD. It's not feasible in most cases. So a little bit about cross ancestry generalization. So this is something that you do have to be careful about using polygenic scores or any type of genetic analysis. Maybe see, like I said, there's systematic there's genetic drift in populations over time that's inherited from if you have a certain genetic ancestry, and that creates issues with GWAS, but also creates issues with the polygenic scores. Most polygenic scores tend to be trained on Europeans, and then if you apply because of the different linkage disequilibrium structure and genetic drift across populations. Those tend to perform more poorly on people of different genetic ancestries or people who are admixed. If you're applying a polygenic score from summary statistics on a GWAS that was trained on Europeans, and you're applying it to people who are of African American, who are of African or East Asian ancestry, it tends to perform much more poorly than if you apply that to a sample of people who are of European ancestry. And so there's unfortunately no good fix for that, but there's something you need to be careful about when applying these to ABCD. One strategy is to do this in a stratified way where you apply the polygenic scores to European ancestry participants, and then you apply it to African ancestry or admixed participants, and then you do a meta-analysis afterwards, or you compare the results that way. Um, So, rule of thumb for genetics and ABCD. So, genetics can be a very versatile and powerful tool. I think especially the polygenic scores can be a very powerful tool in ABCD. The twin component can be a very powerful tool for assessing heritability. But there's issues that you need to be cognizant of based on the design of ABCD. So relatedness is one. You need to be careful about assessing genetic association when you have highly related participants. And then you always need to control for population stratification in any genetic analysis. So those by the way, those are part of the data release. The GPCs, the genetic principal components, are like I think the first 30 or so are included, or maybe more are included as a standard part of the data release. So they're easy to incorporate in your analyses. But that's something you have to be careful about. Well, thanks. I hope this was informative. If you have any questions, please contact me or Chun. I'm sure we can, Angie or others can give you our contact information. Let us know if you have any questions, and hopefully you can use these data and make them a useful part of what you're doing in ABCD. Thanks.